Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology, and now let me summarize what I tried to cover in the last class. That we discussed about the life cycle of the cells, and we also discussed what is the importance of the life cycle in the microbial cultivation system, or in a fermentation process. And we told you that in the life cycle we have lag phase, we have log phase, we have stationary phase, and we have dead phase. Now every phase has some significance. Lag phase tells the acclimatization period, log phase is called the active growth phase, and stationary phase is the starvation phase. Now one thing we should have to remember, that in the fermentation industry we have two things simultaneously we have. One is inoculum preparation, another is the production fermenter. Inoculum preparation, we try to prepare the culture, and the volume of the culture usually 5 to 10% of the production fermenter. So suppose I work with citric acid industry, and the two fermenters we had the capacity of 200 cubic meter, and another fermenter was 225 cubic meter. Now if you consider 5 to 10%, it is around 10 cubic meter to 20 cubic meter. So this much huge volume of culture cannot be produced in the lab, so we shall have to produce in the plant. Now when we prepare this culture, we shall have to see that culture, whatever we inoculate from the lab, it should be in the active phase. And that is why the study of life cycle is very important, because if you in the life cycle you will find that where is the log phase, and usually the cultivation is done in between the mid log phase to late log phase. And then I told you also that most of the fermentation industry we produce secondary metabolites. I have given the example of penicillin in different antibiotics industry, production industry like penicillin, streptomycin. So they are the secondary metabolites, and they produce in the stationary phase. Since the stationary is the starvation phase, we try to extend this stationary phase so that we can get maximum amount of product. After that, we try to discuss the role of batch process and the chemostat. Batch process usually mostly used for determining the life cycle of the cells. The disadvantage of this process that we cannot hold any phase of growth for infinite period of time. So that is the major drawback of the batch process. Suppose if we want to produce the Baker cyst, that the cell mass production maximum take place in the log phase. So since we cannot hold the log phase for longer period of time, then naturally that we cannot get the higher amount of cell mass productivity. But in case of chemostat process, chemostat is nothing but CSTR in the biological system, and in the chemostat process I showed you that mu equal to D. Mu is the specific growth rate, D is the dilution rate, and so dilution rate is basically is equal to flow rate divided the volume of the flow rate divided by volume of the reactor. So volume of the reactor, working volume of the fermenter is remaining constant. If you change the flow rate, you can change the dilution rate. So at different dilution rate you have different steady state condition. So we try to develop the correlation between the steady state cell mass concentration, steady state substrate concentration with respect to dilution rate. Also we try to correlate how the rate of cell mass productivity correlated with the dilution rate. Then we determine that we find out the equation for Dmax. Dmax stands for the maximum the dilution rate, where we will get the maximum rate of cell mass production. Also we come across another point, what we call de-washout. De-washout is the dilution rate when there is no cell present in the reactor, and I try to explain the physical significance of that every cell has generation time, and 1 by d is the hydraulic retention time. If you don't allow the cell to resize in the reactor for longer period of time, the before it multiplies, if you take it out, you will not get cell in the reactor after some time. So this is the disadvantage of the CSTR process, and this disadvantage can be overcome by using the CSTR with cell mass recycling that we try to discuss how this system can be analyzed, and then we also try to tell you how the plug flow reactor can be used. Now, major problem with the plug flow reactor is there should not be any kind of back mixing in the system. So this plug flow reactor can be used for the system where we have the product inhibition, but and this can be replaced by using the multiple CSTR. Now today we will go ahead with this, and we'll discuss some information that is left with us with this CSTR with cell mass recycling. Then I shall discuss the part model and the Ludeking Pirate model, because the Ludeking Pirate model deals with the rate of product formation in the fermentation industries, how it is correlated with the cell mass concentration, and part model usually deals with the maintenance of the cells, and this will be followed by some numerical problems. Now let me start with that. This is the comparison between the cascade chemostat and the plug flow reactor. Again, let me explain that. In case of plug flow reactor, tau plug flow reactor is equal to minus ds minus rs. So if you plot S this way, minus 1 by rs, if you have this kind of, this is S0 and this is S. Now I told you as S decreases, that 1 by rs increases, that means rs decreases, and this is an example of the product inhibition. So in case of CSTR, that the area under this curve will be this, which is much higher than area under for this plug flow reactor. So here the tau plug flow reactor is usually preferable. Now since that plug flow reactor is very difficult to operate, it can be replaced by using the multiple CSTR, what we call the cascade fermenters. This is the, so this is first reactor, second reactor, third reactor, fourth reactor, fifth reactor. So the 4 reactors in series will be equivalent to this plug flow reactor. So the smooth dashing curve represents the progressive decrease in substrate concentration with time spent in the plug flow reactor. To eliminate the product inhibition problem, plug flow reactor is to be selected, but operation and control are very difficult in case of plug flow reactor. For most fermentation process, chemostat offers significant theoretical advantage over the plug flow reactor and the batch reactor. That is why the plug flow reactor, if we replace by cascade reactor, cascade fermenter is usually CSTR in series, then that will be equivalent to plug flow reactor. Now I was discussing about CSTR with cell mass recycling. This is the typical example of activated sludge process where it is largely applied. Activated sludge process, as you know, this is largely applicable in the different chemical and biochemical industries. This the influent is coming, then after this is the fermenter, after that it passes through the separator, the supernatant you take out, and the settled cell mass, a part of the settled cell mass you recycle back, and excess cell mass you take out from reactor. Now here we have, now when we do kind of recycling, one thing I forgot to mention, the purpose of recycling is to increase the retention time of the cells. But because we have come across, you can see that hydraulic retention time is what? That volume of the reactor divided by volumetric flow rate. So this is the hydraulic retention time. Now this is like this. This is the flow rate. Q0 is the flow rate, and and V is the volume of the reactor. Now here V divided by this is flow rate volume per unit time, so it will be time. So this is the hydraulic retention time. So what is the solid retention time? Solid retention time here, the cell mass that is going out of the system, it is not coming back to the system. So actual cell mass loss from the system, we assume this Xc is negligible. If we assume this Xc is negligible as compared to this, then the cell actually what is this? This is the rate of flow of sludge that is going out of the reactor, and this is the concentration of sludge. So you know that QW into XU. QW, what is the unit of QW? QW is the volume per unit time. Am I right? And what is the unit of XU? The cell mass concentration is the mass per unit volume. So volume will cancel. This is mass per unit time. That is what you call. This is called cell mass wasting. Cell mass wasting. Wasting means you are taking out from the system. So this is like this. So now how much cell mass is present? Because we assume here most of the reaction take place only in the reactor, no reaction take place in the pipeline or in the separator. If we assume that, now if the amount of cell mass present in the reactor is what? The V into X. V is the working volume of the reactor, X is the cell mass concentration. So this is the total amount of cell mass present in the reactor, and how much cell mass you are taking out per unit time, that is QW into XU. So that is why the solid retention time is equal to what? V into X divided by QW into XU. That so this is the main purpose of recycling is to increase the solid retention time. We want to, because if you don't recycle, then that retention time of the cells and retention time of the liquid will be same. The hydraulic retention time is equal to the solid retention time. But as soon as you recycle back the cell to the reactor, the solid retention time will increase. The main purpose of recycling of the cell, you want to increase the solid retention time. So that is exactly we have written that in case of CSTR, the hydraulic retention time is equal to solid retention time, but in case of CSTR with cell mass recycling, solid retention time is much higher than that of hydraulic retention time. Now let me discuss about the Ludeking-Piret model. This is very important. The Ludeking-Piret model can be used to indicate the product formation kinetics that combines the growth associated and non-growth associated contribution. Now let me explain that. This explain in details what do you mean by growth associated and what do you mean by non-growth associated product. Before that, let me explain that Ludeking-Piret model. Ludeking-Piret model is the dP by dt. dP by dt is the rate of product formation equal to alpha. Alpha is the growth associated coefficient, dx by dt, and beta into x. Beta is the non-growth associated coefficient. Now if you divide by 1 by x, I told you previously, as soon as you divide dP by dt by 1 by x, we call it specific product formation rate. Now this is when dx by dt is divided by 1 by x, we call it specific growth rate. So this is now specific product formation rate will be alpha into mu into beta. The beta is constant, alpha is constant. So so we can easily plot. We can plot QP into this is a straight line equation, and if it passes through the origin, then beta value should be equal to 0. And if the beta value is equal to 0, we call it growth associated product. I shall explain you how the growth associated, why we call it growth associated product. Now in case your plot is like this is your intercept will be beta and slope will be alpha. In that case we call it mixed growth associated product. Now but we have non-growth associated product, that is particularly in case of secondary metabolite formation, that we have non-growth associated product. In that case what will happen, that alpha will be equal to 0. So then dp by dt actually equal to beta into x, because it depends on the cell mass concentration and it will be a more or less a straight line that we have. So this is, if you plot with x, it will change with respect to, then you can easily find out the value of beta. Now let me explain you how you can define the growth associated product and non-growth associated product. Suppose in a fermentation process, you suppose this is, let us consider this is the batch fermentation process. Here you take the substrate or media and then you put the cell mass here, and time to time you monitor that the cell mass concentration will increase like this with respect to time. Now if your product formation also increase like this, now you see this is product and this is cell. If the product is other than the cell, then it also changes like this. In that case I can write dp by dt is proportional to dx by dt. That is exactly what is called that product growth associated product, and this is equal to alpha into dx by dt. Now in case of non-growth associated product, what is happening, when your organisms come almost in the stationary phase, then your product formation start. This is like this. This is P dash, this is the product. So this is an example of non-growth associated product, because rate of product formation obviously it proportional to the cell mass concentration, but this is occur when cell mass that attain the stationary phase. So this is equal to beta into x, and this is here the example of the non-growth associated product is your different antibiotics fermentation process, citric acid fermentation process, and growth associated product we have the alcohol fermentation process. But the anaerobically we convert the glucose into alcohol. So this is an example of growth associated product. Next let me discuss about the part model. Part model is previously it was assumed that all substrate entering into the cell is used for the growth only, but practical purpose the substrate used for the cell has the maintenance function. That means whenever we use some kind of substrate, a part of the substrate goes for the cell mass formation, a part of the substrate goes for the maintenance of the cell. Now what do you mean by maintenance of the cells? Maintenance of the cells means cells has some motility, because in the liquid cell move from one place to others, and this motility of the cells you require some energy, and so this is one how the maintenance energy used. Not only that purpose, for the cell ruptured, because due to some other reason, due to shear force and other things, the cell may rupture, you have to repair the cell, you require some energy. So all these different protein formation also we required some kind of energy, that is we consider as a maintenance energy. Now if you the part equation say like this, the rate of substrate degradation is equal to rate of substrate that is used for the growth and rate of substrate that is used for the maintenance of the cells. Now I already explained you. Now if you write ds by dt minus ds by dt, what I can write, this is equal to ds by dx, and this is dx by dt. Am I right? Now if this dx by dt is nothing but 1 by x, this is called yield coefficient, and this dx by dt is equal to mu x, that we already did it in early class. So we can substitute this minus ds by dt overall we can replace by mu into x y x by s overall mu into x y dash x by s. Now here I want to point out like this, here the rate of substrate the overall that consume overall substrate consumption is equal to the substrate consumed for the growth of the cells and substrate consumed for the maintenance of the cells. Now this we can write like this for the growth of the cell, this is overall growth of the cells we can replace like this mu x by y x by s overall, and this is growth we can write mu x y dash x by s. Now y dash x by s is considered as true yield coefficient. This is y x by s dash is called true growth yield. Or growth yield, or we can write it the cell mass yield. That means this is equal to also dx by ds, same as overall that overall yield coefficient, but here this ds by dx means the per gram of substrate how much cell mass produced when all the substrate is used for the growth of the cell only. No, it is not going for the maintenance of the cells. That is called the true yield coefficient. This is coefficient Cn. This we can write this. Now another thing I want to tell you that rate of substrate used for the maintenance of the cells, this is proportional to what? This is when ds by dt, if you write this is used for maintenance. Now this is proportional to the cell mass concentration. More cell mass concentration, more will be the maintenance. Less cell mass concentration, less will be the maintenance. That is why when you equal this is m into x. M is the what you call maintenance coefficient. This is called maintenance coefficient and unit is time inverse. This is called maintenance coefficient and unit is time inverse. So here interesting that if we plot now, if we plot 1 by y x by s overall, here we can plot 1 by mu, you will get a straight line plot. The slope will give you the value of m, what you call maintenance coefficient, and intercept will give you 1 by y x by s dash. This is the true yield coefficient, true growth yield coefficient. This is like this. We can easily find it out that for the cells. So this is how we can find out the part models. By part models can be used for finding out the maintenance of the cells. Now let me discuss some problem. First problem that we have chosen, that is Pseudomonas. You might be aware that about the Pseudomonas is very this is the bacteria which was first genetically modified and is used largely in the wastewater treatment process. The Pseudomonas that has the minimum doubling time is 2.4 hours when grown on acetate in a chemostat operation that follows the Monod model. Now K's value is 1.3 gram per liter, yield coefficient 0.4 gram of cell per gram of acetate. S0 is the initial substrate concentration is 38 gram per liter, that means the initial acetate concentration is 38 grams per liter. So what you have to find out the value of S and X. X is the steady state substrate concentration, X is the steady state cell mass concentration at dilution rate D equal to half Dmax. Dmax is I told you in the last class, Dmax is the dilution rate when rate of cell mass formation is maximum. And also you have to find out the cell mass productivity. What is called the cell mass productivity? This is how much cell you are producing per unit time, that at 0.8 into Dmax, and you have to calculate also D washout. So this is a very interesting problem. Let us see how we can solve it. Now you can remember that that we calculated the doubling time. Doubling time, how you calculate dd equal to ln2 by mu. Am I right? When you use the minimum doubling time, then this will be d mu max. Though in this problem, it is mentioned that Pseudomonas has the minimum doubling time 2.4 hours. So if it is 2.4 hours, then we can easily calculate mu max equal to 0.28 hour inverse, 288 hour inverse. Then d max you can calculate with the help of this equation. This we derived in the last class. Mu max 1 minus root over ks plus s0. This is equal to 0.23 hour inverse. Now in this first part you have written, they have d equal to half d max. So we can have dilution rate equal to 0.1178. Now we have also find out that steady state, under steady state condition, the substrate concentration equal to Ks into d, mu max minus d. So if you put this different value Ks d, you will get the substrate concentration, that is 0.899 gram per liter. The x you can calculate y x by s, s0 minus s, and this is equal to 17 gram per liter. So it is very easy to determine the steady state substrate concentration and steady state cell mass concentration in the reactor. Now in the second part, it is written dilution rate is 0.8 into d max. D max value already we calculated. So your dilution rate is what? 0.188. Now what is the cell mass productivity? Let me show you. Cell mass productivity is what? Cell mass productivity. Am I right? Productivity is what? Usually it is the whatever cell mass concentration is there multiplied by d, because we have shown you this is x, d is under steady state condition and sterile feed d equal to mu, and this is will be equal to dx by dt. dx by dt is the mass of cell produced per unit time. So this is exactly that cell mass productivity is equal to x into d. So we have this dilution rate. So you have first we shall have to find out what is the cell mass concentration at this dilution rate. Then we can multiply that x with d, we will get the value of the cell mass productivity. This is exactly they have found out that x we find out that 16.35, and then you multiply it by 1.188, we get the cell mass productivity whose unit is gram per liter per hour. Now d washout again is simple. Had d washout the x equal to 0. Since x equal to 0, s will be equal to 0. So d washout equal to mu max s0 ks plus s0, and this is equal to 0.279 hour inverse. So this is the show we can easily solve this problem. Now we have another problem with us. The steady state substrate and biomass concentration in a chemostat operation is given by S0 equal to 700 milligram per liter. Calculate mu max and Ks, growth yield coefficient, true yield growth yield coefficient, and the maintenance of the maintenance coefficient. So you know that this is not a batch process, this is a continuous process. So it is a chemostat operation. So they have given the different dilution rate, what is the steady state substrate concentration is given there, different steady state cell mass concentration is there. Now in this equation, if we use the Monod equation, we can write in the form of Lineweaver buck plot mu equal to d. Then I can write 1 by d equal to ks by mu max, 1 by s plus 1 by mu max. Now if you plot 1 by d versus 1 by s, this is constant and this is constant. From the slope you can find out ks by mu max, and intercept we can find out 1 by mu max. So this is exactly we have found out here, that mu max we calculated and ks we calculated. Then according the part model, that 1 by y by x by 1 y does x by s plus m by mu, and in the CSTR under steady state condition mu equal to d. So I can write this equation like this. Now in this equation, that you see that d is the dilution rate, and then this is the substrate concentration, this is the cell mass concentration, and from that we can easily find out the yield coefficient. How we can calculate the yield coefficient? Yield coefficient is nothing but dx by ds. We know the initial cell mass concentration, we know this final cell mass concentration, and initial substrate concentration and final substrate concentration. From that we can find out the yield coefficient. Then 1 by y x by s overall we can calculate. Then we can plot this, and from the slope we can find out the maintenance coefficient. This unit is our inverse, and from the intercept we can find out the true growth yield, that is about 0.5. So this is very simple. Then other problems that we have, that I have given as a assignment. If you this is for this I shall solve you when I shall discuss your Baker's fermentation process. Thank you very much.